Okay, good afternoon, everyone. We are back to what we have discussed in the first of the classes during this course. So we will use this for a similar interaction, where we started and after learning and where we are. That's how you should be able to recollect whatever we have learned so far, and then suggestions, opinions, and other things for the future. That is how we go about this class. So the objectives, I think, are pretty clear and simple. We do not have much in the sense basic concepts and genetics. One should be able to understand how the genetic information is causing a whole cell and how it is training or how it is regulating the whole body itself. So the approach is informational in approach. It's not material in approach, rather it is informational. In the sense we discuss it as if it's an information, and that information gives out or dictates a structure. So we had in the first unit, when I'm talking, it's also important that you note down probably what are the important things. We should learn about Mendel's discoveries, especially the ratios, and how to draw a Punnett square and so on. And most important of these would be about the genetic material, structure, the chemistry of genetic material, DNA, RNA, and also the experiments. Right? There are 3 or 4 experiments. You can always go back to the videos that we have discussed, and then you can always have a look at them. And the first unit also comprises some of the techniques, where the techniques are general basic techniques. Probably not in-depth questions would be asked in it, because if you finish the lab course, it would be much better. But anyway, it's as a student of molecular biology, one is supposed to know them. So we discussed it. You can always go back and try to learn those. In Unit 2, we learned about the genomic organization, types of DNA sequences, nucleosome, chromatin organization, and so on. And then we also learned about the replication of DNA. So you can, if you are looking at as a essay question, it would be describe the genome diversity or chromosome diversity, types of sequence diversities, or the organization of chromatin. Similarly, you can say in replication, one would learn about the chemistry of polymerization, the enzymology of polymerization, telomerases, telomeres. That is one important thing, and something like PCR, right? So the questions, if it were a normal regular course, questions could get a little more complicated and so on. But at this point, it will not be too complicated. At least that is how I intend to put the question papers. And in recombination, simple, pretty straightforward. There are 3 important things: homologous recombination, and we have site-specific recombination and transposition. In all the cases, we should also, the recombination part is one thing, and the repair part is another one. Types of DNA damage and types of repair mechanisms, right? So if you go through the PowerPoint or the figures and legends in the textbook, for all of this course, it will be more than sufficient, actually. And I will describe about the model question paper, then you probably will understand what I'm expecting when it's time for you to answer. Then we have, in the next unit, we have, in 4th unit, we discussed about transcription and translation. In between, we also have about splicing. That's an additional chapter. And we have discussed in quite a detail all through the course. I will get back to one point. I want to verify or elaborate on one point. The difference between, as a bachelor student, the difference between majority of the institutions in India and abroad. is there is in abroad, what I have observed is there is no concept of notes. There is no concept of studying from PPT. People study textbooks. And that is what I tried my best to inculcate by following a said textbook, using the same material for discourse as well as for making PPTs. And I tried my best to also make you buy the textbook. And if you have followed the instructions, you are learning it in the most authentic way, that is, reading the textbook. Eventually, your ability to comprehend the textbook is what I wanted, right? So we covered in great detail, not the entire textbook. It's not possible to cover the entire textbook in one course like this, not especially not online, of course not. But even then, if you understood whatever we have already discussed, and you can always read the textbook and understand more and better, then you are successful through the course, right? A textbook typically is read for the first time only after reading for something like 2 or 3 times. That means it's not entirely understood right away. We don't retain everything in our minds. It is volatile, and that is natural and common. We will forget. Right after this course, I might forget some things here and there. I will remember the principles or the logics of some of the phenomena, which I find interesting or something like that. Not necessarily every bit and gene name or protein name we cannot remember them all. But for the exam point of view, from the point of view, you have to read. So even if it is short-term memory, try to read that and try to memorize a few things. And in gene regulation, that is our fifth unit. We have learned about gene regulation in prokaryotes. That was our major focus. And we did learn about some things about the eukaryotic gene regulation. Not so much in detail, but we still covered quite a few topics and some of the pathways. So at least the least expectation is that you should have known the regulation. You should have known what it means. What does a word, something like imprinting or epigenetic gene regulation mean, right? And once you know the meaning of the words or the terms, then everything else is very easy. Okay, so that is where your memory should come in. Try to know the terms, the meaning of the terms, and then the rest of it will be easy and fine. Okay, and these are the learning outcomes. I hope you will agree that we have covered a lot, and I'm through the assignments. Okay, my assignment, when I give the assignments, my objective is that you should be able to fulfill these outcomes. Now I think you are able to do the Mendelian type of these Mendel's questions or problems based on that. You know structures of RNA, DNA, you can draw them. And then some of the principles, that's what unit 1. In unit 2, you will be able to explain. Okay, those are the minimums that we are expecting from you, is explain. And then you probably can take it to higher level. Higher level will be application, creation, and so on. But because this is a BTech or BTech second year course, we will at least keep it in understanding level and a little bit here and there. But otherwise, majority of the things are understanding level. Okay, so what I now want to do is to take you through a question paper model. And there will be four questions in the exam, and each is an either or type, in the sense that you will have one question, and so you can have one question, and question in that question A and question B will be there, and this will be XYZ, and this one could be PQRS, and you will be asked to choose to write either A or B, and each of them will cost 20 marks. Okay, And then you also have, yeah, so each is 20 marks. So you'll answer. And in Code Tantra, when you're doing it, you will either see the first question or the second question. I mean, you'll see both the questions, both the, for question number 1, A and B will be shown. You can choose to write either of them, not some parts from there and some parts from here. That's not the case. You have to choose one of it and write. They may have sub sub questions. In that case, if there are sub questions, the instructions, current instructions that we have received for the other exam says that there should not be more than 2 sub questions, and the division of marks should be equal. So you have, say for example, question 1B. In that we may have 2 bits like this, and each of them will be 10 marks and 10 marks. Okay, so 4 questions, 4 into 20, you will get 80 marks. For the final grade, it will be converted to 75 marks. 80 marks will be converted to 75 marks. And internal marks, you have about, for 25 marks, you have done assignments, which is 4. Okay, 5 marks each, you did 4. So you'll get 20 out of it if you have done it all. And 5 marks is for the compulsory assignment that we have given. So that is for 25 marks. I'm glad that majority of you have scored well and did fine. And the model question paper is something like this. One question will be sequence analysis, which is also the same as what you have done for the compulsory assignment. You will be given 2 sequences, and you have to find the differences, calculate whatever is asked. It will be very much similar to what you have done for the assignment. So 20 marks are avoided for sequence analysis, awarded for that. And majority of people, I mean, I found that in the, for the compulsory assignment, not many people have chosen. I mean, some 8 or 9 people did not choose to, they chose not to submit it. And that's, I don't think that's a good thing, because now you should practice it, because that's a question that's going to come. But otherwise, questions will be like this. Something, for example, in based on Mendel's laws, a question will be given, which is a description of a problem. And you should be able to say, if required, you should be able to draw a Punnett square and arrive at the ratio. And based on that, you should be able to tell, say, the question is about calculate the number of plants that are genotypically homozygous. So you reading the question is very important. Reading the question properly is important. And also reading it and also understanding the words. If you didn't know what is homozygous, there is no point in how you would actually answer, or you wouldn't know what to answer. So keep that in mind. And the second part, so the first question is 10 marks. First part of the first question, question A. So, and the second part is, I asked describe about Griffith's experiments. In that case, you should be able to describe the objective of the experiment, results and explanation of the results, and the rationale, and so on. So there are 3 or 4 experiments that we have discussed in detail. You should know those steps, or that is how you are supposed to present the paper, answer for it. So chemistry of nucleotides is very important. I might ask a structure of a nucleotide, dinucleotide, or a base pair. You should be careful in what you're answering, how you're answering it. And you would also say 5' G cap, and many structures are there that are of interest. You should be, you should know those structures now itself. And it should have the labeling properly. Labeling, and with the 3' groups, what is the names of the bonds, and so on. Just your ability to draw that is giving you 10 marks. Okay? So try to remember those things. So this is another structure, chemical differences between DNA and RNA, and so on. DNA replication, second question, for example, DNA replication, which is by semi-conservative method. So you should know the biology, chemistry, enzymology of it, and that description. And everywhere in all the questions, you will have something like this. Wherever applicable and possible, you should try to rely on labeled schematic diagrams. I can tell you now itself that you should have practiced the diagrams beforehand so that you'll feel comfortable during the examination. If you just watch it, you wouldn't know until you actually put the paper onto, I mean, pen onto the paper, that it is not difficult, it is not easy. If you have practiced and made up your mind, okay, this is how I will draw, and you practice it with an experience, then you would know exactly how much time it would take, what are the important things that you should focus on when you are drawing a diagram. Okay, diagram is the best way and easiest way to display how much you know. Okay, so keep that in mind. There is no limitation of your language. You don't have to search for words or making proper sentences. If you could draw it, then it is fantastic. But ensure it is, they're all labeled schematic diagrams. So in question 2b, then I could ask about cell division. We did some problems based on that, meiosis or mitosis. And the other part of it is also about the chromatin organization, right? So it is 20 marks. And then you will have to do some of the things, calculations and so on, and diagrams and description. Diagrams itself is having 10 marks there. Sometimes you have to draw the diagrams, without which you would not be able to explain properly. This is a question which is a little bit thinking. Thymidine dimers, you should know the cause of what would have caused the thymidine dimers. That's UV light and something like that. Cyclobutane rings, you should be able to say, and then how it gets repaired and so on. So I hope you see that the questions are pretty straightforward. If you understand what is happening, then why something could happen or what will happen because of some other event, then you can always answer it properly. Here is an example. Here is a question with some requirement for specific details. Compare homologous recombination and site-specific recombination with simple illustrations. You know what is homologous recombination, you know what is site-specific recombination. Then you should be able to say what are the similarities and what are the differences. There are very little similarities between these two, and there are a lot of differences in the mechanisms, outcomes, and so on. So you should try to know that. And the second part of it explain the regulation of Hin recombinase, which is about site-specific recombination. Okay, in flagellin regulation we have seen, so you will have to describe that. So in the next is about, remember this, we have 5 units and the number of questions allowed is 4. So there will be clubbing of some things here and there, right? So say, for example, you can think of, well, we'll go into the next chapter and there we will see. Question 4a, explain the promoter initiation. And this is purely asking you for the, asking you to just write the about transcription. From initiation to elongation and also to termination. That's a whole thing to write. If you have to write it, it will take a very long time. Instead, you should find ways to explain them in diagrams. If it were in diagrams, you will have maybe 4 pages and maybe 15 minutes or something to draw. But if you have practiced it, okay, so keep that in mind and try to do your best. 4b is differentiate. This is my favorite question always. Differentiate the mRNA of prokaryotes and eukaryotes. You should tell how they are processed differently and what is their composition, how is it different, and so on. The elements, say G-crab or polyadenylation tail, and so on. Okay. And the second part of it is translational initiation in prokaryotes and eukaryotes. So you can again do the comparison between those two. How is mRNA recognized? What is the difference in the ribosome binding site, and so on. So these are some of the questions, that is what is written, and those are some of the expectations that we have when we look for the answers. Okay. So if you have any questions on this, I would like to take them immediately, and then I want to talk about something else, about general talk. Any questions and based on the question paper? Okay, if not now, you can always come back and ask, and then I will try to see what I can do about it. So I always want to start my course with a discussion on careers, opportunities in life. I also want to end the course with same topic, right? And we haven't been in the same, building or physical classroom. So I would still want to use this last class to discuss those things. Let's talk. We are not in academics anymore, or not in molecular genetics. We're just talking about general things. So relax and try to see if it makes some sense to you, or you can come back with questions again. I hope you remember this slide, or I'm unsure if you have seen this or you remember this. We are all studying hard to get a good job, to get more money, to get fame, maybe, to get love or friends, and to get respect. Job is what gives us all these things: money, respect, and hopefully something interesting to do. And there is no shame in saying that money is the most important one. That's the truth of it. If you don't have any of these, it's still okay. Money, without money, there is no existence. That's how human society has become. And that's the truth of it. There is nothing wrong about it. So if somebody is pursuing money, there is nothing wrong about it. It's all right, as long as it's legitimate. And the problem comes is, as a student, I was worried about what kind of job I will get, whether we'll get a job or not also. Okay. Then the things are different. Our dreams and hopes are different, and the realities are different. You are in a good place. Your probability of getting a good position is very high, and you earning good money and being in a respectful position in society is also very high. So that's a different issue. And when we want to choose a job, and I'm talking about the core company type, okay, software job is always there, TCS or some Infosys, some jobs will be there. You can take them up, and then you will you'll always get a job, and you will feel you can be happy about it. What I'm talking here is about if you want to do a job that is in life sciences. Okay. So there are two jobs: industry and academia. In industry, you have research and development, R&D they call it, and there is something production. These are responsible for the development of drug or improvement of the drug. The second team is those are the people who are responsible for operating tanks or fermenters and so on to produce the drug in large quantities, in purified quantities. There is also QC, or quality control, to check if the product is pure or not other whether it conforms to standards and so on. And the management is to ensure that the whole company or the industry or the plant is running properly or not. And then we have marketing. That is, the product is made, developed, made, and quality checked, and now you should be able to sell the product. And for that, you have marketing personnel and team who will popularize it and try to spread it across so that they will they will be able to produce. So one can get a job based on any of these things. You can be recruited into any of these. What you are trying to do, where you want to be, again, is a question of what you want. Okay, say in research and development, it is very challenging. It's more about brain thing. You have to study mental work. Pressure is everywhere. Okay, there is no job without pressure. Even Tendulkar, if he is playing, and sometimes it's not always fun. It is many a times it is pressure, because if you don't perform, it is going to be it might be, you might have to face some uncomfortable comments and other things. Here in research and development, it's mostly employing the brain. In production, it's typically, there is brain work, but it's typically repetitive. And this quality control is also repetitive, because you have your product will come, you'll check, and it's a kind of routine work. And management is, I wouldn't want it to do that, because I don't want to deal with people in the sense that to tell you do this, you don't do this, hey, why are you doing like that? This people management is actually a difficult one. And so if somebody is good at it or likes it, they should opt to go for management work. And if one becomes good in either of these, they would also get into the management ways. It's because they know the bads, and they will be able to manage. If somebody troubleshooting, if somebody has a trouble there, they will become good at it. So eventually, as people grow, management will come into the job responsibilities. And marketing, if you are a traveler, you like to deal with people again to sell them a product, then your skills will be in marketing. Entrepreneurship is something you can own your own startup and try to make your things happen. The probability is usually lesser, but we have in our own alumni, there are a few, at least 2 or 3 people who have tried, and some of them are actually successful in trying to have their own brand of equipment and so on. The other part is academia. Okay, that is where you have 2 responsibilities. It depends on which level you're entering, where you're entering. Typically, everybody's base responsibility is teaching, and everybody's recognition comes mostly from research, because when you are teaching, you are confined to the classroom, in the sense your work is recognized by the class alone. But when you do research and publish it, okay, you are getting the whole globe know about you. And that's why it is important that one should have, if you are planning to go for research, teaching, you should plan to go for research too. I'll get back to this slide in a while, so that, not this slide, but a similar slide, why I wanted to discuss that. We are all in this situation like this because of COVID-19. And we always, I think at least some of you wanted to be the doctors. And doctors and the personnel are helping cure and take care of the societal, this pandemic in general. But we have an opportunity to support doctors. Where did the medicines come? Where did diagnostic kits come? Where did they come from? They're all developed by scientists, engineers, technicians, and they then are those products are developed and given to the doctors and the hospitals, and then they try to do administer these technologies on patients, right? So this, if you ever wanted to become a doctor, I wanted to become a doctor, I couldn't become a doctor, and then I wanted to next become the next best thing for me is, or it is in a way, why did I want to become a doctor? Because that's the most respectable one, respectable profession, because you are dealing with life and death situations, right, on a daily basis. As a part of your regular job, you're dealing with life and death situations, and that's why it demands or commands as much respect. So if not that, then this is the next best thing that I would want to go. Generate knowledge, generate diagnostic kits, manage things, and produce several other things, treatment strategies, how to prevent, how to manufacture different things. And if you still are caught up in, I should have been a doctor, but I'm not a doctor, then you are in a good position and a good program to study further and be above doctors. There's nothing like above doctors in the sense you are trying to develop something that can be used by doctors in curing the disease. Right? You're not directly administering. We don't have license for do it. Only doctors have license to do that. And we are reinforcing the doctors' tools, right? Tools, techniques, medicines, and everything. And that's how you can still be of great value to the society. As bioengineering students, you have a vast diversity. You can think of any of these issues, something like knowledge, diagnostic kits, prosthetics. You can think of many, many things, and you can be useful to the medical field or any other field in general in a very diverse ways. So you should try to look into it again, and try to see what you would want to become. So for me, this is where we are going into science. That is, it's called sciencing. Sciencing is a verb, right? I am sciencing means I do science. That's what it is. That is, science is, you will find out many, many things. How do I get into science is one thing I want to talk about. But at the same time, I also want to talk about, it's a job, right? When we talk about it as, and I'm putting it here about money and respect, and I'm trying to go through the path of our education, and then what jobs might come up, or what level of job would come up. So time and education level, and we are in school. People can go to, from schooling itself, they can go, they can get out of the path of education and go to jobs. But then they will be at a very level, they will be at a very low level entry, and their growth is limited to a certain level. You cannot have more respect and more money than that. There will be exceptions, but in general, that is how things are. After bachelor's, BTech, or mostly BTech and BSc or other things, also you can study further, and then some people choose to go for jobs. You would have a start and stop, and that will be the next level of your money earning and respect. And then master's levels, my suggestion is that you all should have a master's degree. That's the kind of minimum nowadays. Okay, so BTech alone is insufficient. But after MBA also, people can choose to go out of it. And then you have the ultimate, or kind of, okay, formally, if there is a final degree, it's PhD. And there are Doctor of Science and other things, but we'll just say PhD is the tip or the formal end of education. And then also you can go. So in each case, you have to spend more time right? In each case, you have to spend more time, more efforts, more focus, and some money to get to keep studying. Until here, you have to spend money maybe until master's degree. For PhD, usually you are paid. It's a kind of between education and job. You get paid, but you have to go with some fellowship and so on. While I have said these things and I drew a graph like this, it is not an absolute one. Okay, it's you can finish a PhD, and you if it is a bad one, it doesn't mean it does. It says that you cannot maybe enter into a job at all. Okay, so or I shouldn't say bad probably or substandard PhD. Sometimes people just get PhD just like that. So what I meant here is if you are doing it in a very good way, and and the standard should be good, standard of the quality of your whole degree should be good. I hope I gave you some ideas about what you want to decide. You are somewhere here now, and whether you want to choose to go for a job or you want to go for higher education should depend basically on your income, on your interest. Okay, and the other one is income. Say for example, if you after BTech somebody offers you like Google or somebody, they crazily they offer 50 lakh per annum. I think it's all right to take up the job as long as it is comfortable for you, right? And if somebody is offering you 3 lakhs per annum, there is no point in taking up that job unless you have a family burden to take up immediately. If the family needs a help or they cannot afford anything else for you, those decisions will be dependent on what is possible in your immediate with the family financial status and other things. And I'm assuming that nobody typically, it's unusual that we will have financial troubles so early on. So my suggestion would be to study as long as possible, because we need to grow, right? And we need to keep growing. Your education from here till here will make you a student forever. That means you will keep on learning for the rest of your life. And that's a very important trait anybody can have. So that's what is your career paths. I hope you will try to reflect on it, and you can always have dynamic plans or no plan at all, and only a goal of learning, and then it will take you a long way by yourselves. Whatever you do, do it well. That's the important thing. The factors for success are, those are the two factors. Where you study is important. If you study in a substandard university or college, then your degree, value of your degree and the quality of education may not be good. May not be good. And the second one is how well you study. If you have worked hard, and if you have worked hard in a very good place, it doesn't matter what you study, right? You are a person of knowledge, you're a person of higher cognitive skills, you're a person of ethics, and you will just do it well. And that's a good, it increases the probability of your success. So why does the place matter? Is because you need to have an ambience of knowledge, resources for your growth. It should not be, the institution should not have a limitation on your growth. Institution should be much more broad and wide, so that when you spread your wings, it still has space for it. It's not like there is no space, so you should cut down your wings or something, right? And you should have, of course, a lot of encouragement. And usually when you have space and a heart, then you can probably, the institute can encourage you more, right? When it has some things to give, it will encourage you to take. If the institute doesn't have enough to give, then what's the point of encouraging, right? So that's the idea that I have on that. And of course, how well you study is important. And my biggest one is, if there is a skill that is called self-reliance is the skill, and always try to study well. Even next year you will have fundamental courses, immunology and other things. Try to read them up. Try to read your biochemistry and other things: biochemistry, cell biology, molecular biology. These three will form the basis for anybody who is interested to do research in core biology. So keep that in mind. So I was telling you about academia and so on, right? It doesn't matter if you are a doctor, engineer, or scientist. Eventually, I would want that everybody, those that have gained enormous experience in real life, with who are dealing with the actual scenarios, they should engage in teaching. Because if I don't know, if I don't have experience in what I'm teaching, my level of teaching will be different. It's not the same commitment. It's not the same level of wisdom and understanding of it. I love molecular biology or recombinant DNA technology because I practice it as part of my research. Okay, I deal with these sequences, and that kind of work is what I do, and that's why I can associate a lot more with that. But if it were, say, cell biology, for example, I'm not teaching cell biology. I'm not dealing with cell biology in my real research, and it's not the same way I can teach those that are working with cell biology, right? So I think when you become a successful anybody, say engineer or scientist or even a software engineer, try to see if you can become a professor and get into academia at some point, not immediately also. Because I think in Indian system, the problem is those that are teaching are not practicing or didn't have enough practice. I'm talking about normal, say, B grade institutes or something like that. So the teacher should have been engaged with actual practice the science of what is being taught, and that will be the best way. That is where in academia, when I wrote in the previous slides, teaching and research should go hand in hand, and that way it's good for the teacher and great for the students. And research is fantastic because that can get you a Nobel Prize. It's far, but it is within reach. All we need to do is work a lot more. Keep working until you get one. Be in great places so that you can do great things. Small places can also do, but the probability is little slow, right? So keep that in mind and see if you can get into research. Do a PhD, as long as there are no financial issues. So what is research is, it is for me a philosophy. It is trying to find the nature, the truth about something, right? You have to apply, it's an application of knowledge to generate more knowledge. Everything is, eventually everything is knowledge. We start, first knowledge is language, and then every other knowledge flows through the language into us. So it is a great thing to be in that. It's not easy. It's on a daily basis challenging and daily basis thrilling in the same way. There is not much of routine in there, okay? Because the problems keep changing every day. You can study at any level, okay? If you are into research or want to get into research, you should think about, do I want to work at the level of atoms, or do I want to work at the level of DNA or nucleic genetic information, or cells, or bodies, or groups, or communities, or population? You can choose any of these. And here I have listed some areas that I find something like what you will also fit in. Molecular biology, and all these are very important. Everything is important. It doesn't matter what you do, as I said. What matters is how well you do, where you do. If you did it in a great place with very you did very well in a great place, then you are successful, right? So it doesn't matter what you study. That's my opinion. And then one second, yes. So I will, this is one important slide, and then probably I'm nearing to stop. This is us, right? And we have input, and then we are the processing ones, and we give an output, right? What is, and the devices required to take in and put out are all the languages. What languages? It can be English or mother tongue, anything it can be. And in between, we have we. And this is the one that has to do with some things like thinking, or actually the whole of processing. And processing includes thinking, thinking properly, thinking and executing in or deciding based on integrity. I mean, that is honesty and professionalism and so on. And say self motivation, self awareness, thinking, integrity, and professionalism. These are all attributes of this person, right? And of course, the input is, and while the input is more, we keep accumulating knowledge. And our speed and with which we work and all that will give us intelligence, creativity. All these things are attributes to us. So if we want to do better, we should first have languages, because that is the door that will allow the input of knowledge. We should be able to read consistently. We should be able to find, we should have the knowledge, energy to find the knowledge, right? The resources for information. And then we should have proper critical thinking skills, system thinking skills, or design thinking skills. Of course, we should have had knowledge based, and knowledge doesn't come just in a blow, and it's not like a computer is loaded with some files. It is a gradual process. It happens from our childhood on. So most, if you are successful, the elementary school teacher, including your mother, are the parts of your success. We never talk about our elementary school teachers or kindergarten school teachers. I think those are the people who have, apart from home, of course, mother and all, they have opened the doors for us to gain these skills. And that's why I think that's the most important part of our successful, for a successful life. And output, if you want to say, what are the skills for output? You should be able to write, right? You should be able to type right. Say, for example, you are going to make reports in the, for even for lab manual, lab book, or lab record, or eventually in your project reports, you have to type. Did you learn about touch typing? Right? Do we know touch typing? If we don't know touch typing, how difficult it is to make a document, isn't it? And does it require so much of energy? No. Do we have the resources to get ourselves trained in touch typing? Yes. All you need to do is either online or offline. You can download some programs and practice typing every day. And if you get this touch typing skill, then you will be able to do it much faster, the output. You will be able to do effectively. And do we have skills for that? While it is available, why aren't we doing it? Is the other part. So The success just does not depend only on this. It also depends on our skills in ability to acquire more knowledge, input of knowledge into us, and also communicate the knowledge that we have already gained. That is the output. We are just the processing machine, and we should be happy. And for that, I think we have to improve our skill sets. Programming is going to be essential for everybody. I never learned programming, and now I'm trying to learn a little bit, and it's not that easy. You people already have a lot of introduction to it, so my suggestion is try to learn uh, programming, become an expert in some things. Okay. So these are some simple things. Respect in life, money and power. I don't want to talk about it, but it is about what kind of information do you have? How authentic is the information you have? What quality of information you have? Second one is what kind of skills and expertise do you have? And the third is our attitudes and ethics of work. Are we energetic to say, okay, I will take on the work, or are we a good lord again, work? Why? That kind of attitude. When we go for a job, our respect will depend on these things, right? The society will respect only based on our money and other things, but where we work, where in our peer group or the institute where we are, immediate respect comes from these things. The quality of us, the quality of the person, that depends on information, skills, and attitudes. And I hope you will all find a good job, because employment is nature's physician and is essential for human happiness. And the happiness, you should be able to either you're doing what you like, or you're liking what you do. Either of the cases should be the goal, and it's still not always possible. What you do might not be happy, but you might get very good money, and then some people can say, okay, I can take a little bit of pain, and we do many a times. So try to supplement your life with uh, with with arts and sports, so that you can always live your instincts. We have skills, we don't use them, they're gone, and we are sad. These are some of the doors that will make us feel what we are, and that is when we feel what we are, is what happiness is, because we can live what we feel inside. Say, you can't go and hit a person, right? You can hit a shuttle in badminton, for example, and it's expenditure of your skill. You live your skill to hit something, and in sports you will be able to do that. You want to express some idea in the form of a painting. If you have it, do it. If you want to dance, dance it. Sing, do whatever it is. And those should be the doors for you to keep yourselves happy. And it's the energy. I hope you learned about Simport, right? Simport in membrane transport. So X is outside, it is 10, and X inside is 1. So the gradient is here. And that can be used to take up something else also, which is against the gradient. Say, for example, Y is 3 here and Y is 5 here. This electrochemical gradient could be used to take up Y, right? So that is, one process is driven by another process, and that is how I see where you can use sports and arts to drive your other, say something like learning. Learning is difficult. Studying is difficult. So we can mobilize our own life or keep the motor running using these arts and sports in daily life. Sports more importantly, because you have to move your body to the best of the possibilities, stretch big time. And while doing so, if you get paid, you're lucky, right? So this is the last slide. Anyone who stops learning is old, whether at 20 or 80. Anyone who keeps learning stays young. The greatest thing in life is to keep your mind young. Henry Ford. I love this quote, or I have learned it, that we have to reach a stage where we feel dull when we don't learn something new, right? You can learn some skill, you can learn some knowledge about how a fan works or how a mobile works or something. Experience technology. If you didn't play with the remote car, play with the remote car, dismantle it, find out how it works. That's amazing. So some form of learning. It doesn't have to be academic learning. Every form of learning. You can start poetry today, and while doing that, you're going to learn a lot. You can write a book, you learn a lot. Some form of learning should be there. Don't fall into stereotype that we should be like this and this is where we reached. Now it is time to raise the next generation. That's not the case. You are still growing, we keep growing, and we keep learning until the end of times, right? Okay, I'll stop here, and everywhere try to remember this. There are many people out there who will tell you that you can't. What you have got to do is turn around and say, watch me, right? I hope you have understood my some of the things that I wanted to share with you. It would have been nicer to have been in class along with you, and then it would have been nicer, but this is the best we could make out of this situation. Thank you very much for the whole course. I am really impressed with the assignments, and I hope I'll be seeing you around often. Okay? Thank you very much. If you have any questions, please ask, or you